Okay, let's get started. Before we go to the next session, do you have any questions for me? During this break, I'm sure you discussed something and come up with some questions. Any questions I can answer before we go? Yes, please. Questions are always welcome.
Very simple. I don't know. So, which study is better in vivo or in vitro?
We have to start with in vitro. To say like a lot of the experiment, right? Simple experiments, you can get it done. In vivo takes time. So as we know, right? You create an animal model, takes years and years. So while you are waiting, you can complete all the in vitro experiment to justify why we need animal model. For example, in the in vitro, if we don't see any differences, then how are you going to justify you need an in vivo model? You are putting time, you are putting money, right? And then you are going to ask a funding agency to give money. You need to justify. Or if you don't have any result like showing differences between the mutant and the wild type, you need to justify why we have to create animal model. Unless you have plenty of money, then you can just generate a model and then you can study. But normally the in vitro experiment is the starting point. You can easily get it done with the available resources in the lab and then justify to go to the animal model. Even for our 25 base pair deletion, we first published a paper in JBC just to show all the in vitro experiment. And then we generated a mouse model and then we asked for money. In the mouse model, you show preliminary data with the phenotype and then you ask for money. I want to characterize this model. I want to show the phenotype. I want to treat this disease, etc. Then you get the money and then you do 3 to 5 years time to study the mouse model. And then we published that one in the cardiovascular research, which is 12 impact paper. So you go step by step, again based on how much money you have in the lab. Most of the labs are middle class labs, and so we start with the in vitro experiment. Again, once you know that it's big difference in the phenotype, this is an easy start. But at the end, even though you have an in vivo model, you have to come down to do the in vitro to justify, to give the rationale and what mechanism is involved. Is it myosin heavy chain is activated, or how do you tell? Only by measuring ATPase activity, right? Yeah. Okay, any question? So going to the in vivo model, again just want to see the differences. So you have the normal heart and then the hypertrophic cardiomyopathy where you have more thickened wall, and then the dilated cardiomyopathy or heart failure, your walls, the ventricular walls are very thin and then the chamber size is so big. Pretty much end stage heart failure, you need a transplant. So why are animal models important in hypertrophic cardiomyopathy research? What do you think? Why animal models are? You said, why don't we go directly to why? Why in vivo models are important? Why can't we just conclude with our in vitro model, this is the mutant and this can cause phenotype, done. We can start the treatment.
Because the organism as a whole is composite of various different systems that comes together to
Yes.
form out being.
So it's a physiology, right? You want to see the whole animal level, organ level during the development, how the disease can be prognosed. So importance of animal models, number one, we are replicating the human condition in an animal model. Exactly, we are introducing the mutation either through knock-in or knockout or transgenic overexpression of mutant protein and then mimicking that. So we can study a lot of these properties only in vivo, not in the cell culture experiment. Hypertrophic mechanism, fibrosis, arrhythmogenesis, whether that can develop left ventricular outflow tract obstruction, which is the case in myosin heavy chain mutation, and then heart failure. So again, hypertrophic cardiomyopathy, as you know, have you seen the last 3 days, it won't happen overnight, right? It's not like an infectious disease. Get the infection, you 1 or 2 days, you see the impact. But this takes years and years and years. And some of the genetic mutations, it takes more than 50 years, 60 years. They cause late onset. Some diseases, some variant can cause early phenotype. So definitely the time point in the disease development only can be studied in vivo, not in vitro experiments. So again, when you look into all the pathways in the myocardium or in the cardiomyocytes, there are a lot. We already have number of drugs, small molecules that being used right now. For example, we talked about the beta blockers, the calcium channel blocker, ACE inhibitors. So there are a lot of already been in the market, in the clinic to treat heart failure. Now we have mavacamten to treat hypertrophic cardiomyopathy. So we have to have a preclinical model to test all these various drugs and see whether that can be translated into the bedside. So you start with preclinical model, again you validate everything in vitro, and then you go to the animal model where you can study safety and efficacy study, and then you go to the clinical trial, and then you go to the final FDA approval. Again, it's a very simple diagram, but each step there are a lot of steps to be completed. So animal models of cardiovascular disease. So here I listed starting from mouse model, but even zebrafish, Drosophila, they also been used in studying hypertrophic cardiomyopathy or heart failure, heart disease as a animal model. But again, at least a mouse model, it's very close, better models than the zebrafish or the Drosophila models. Why are mouse models are predominant? Again, you can take all the models, only been mouse models being the predominant model being used in the literature. So number one, genetics are relatively well understood. We know the genes, all the genes of the mouse, well characterized, well known. There are various strains of the mouse. This is the FVBN strain, white. There are BALP-C, C57 or 126. There are so many different strains. Again, remember that these strains can also give different phenotype outcome. The same mutation, if you put in different background, you will have a different phenotype outcome. So you have to again know what you are selecting. So and then the genes are almost the same comparing to human. We can generate the colonies so faster, defined cardiac mechanics and hemodynamics, and then this offers more preclinical tool to study any treatment options or toxic effect, etc. So again, as I said, the disease development can take time. So if you have an animal model, you can really look the heart every month by echocardiogram, right? So you can really look the developmental process by doing echocardiogram. This is a non-invasive. We have a small echo machine that can be used to measure mouse cardiac function, and so we can define at what time point the disease is now starting, So in the literature, if you look at the literature, a lot of mouse models already available. So transgenic are very common in the early time before the CRISPR was introduced, and then we have CRISPR-Cas9 genome editing is now. The transgenic is like cardiac specific transgenic is we use alpha myosin heavy chain promoter and then tie the gene of interest with the mutation, and then you have the human growth hormone polyadenylation sequence to have the stable mRNA in the sarcomere or the myocardium. So all these approaches were used in the past to generate mutant mouse models for myosin binding protein C and myosin heavy chain. The knockout models are being used to study haploinsufficiency. Again, if you have one allele wild type, you don't have the second allele, this one allele is enough to produce 100% maximal protein when you are young, but aging process can cause haploinsufficiency or presence of any secondary risk factor. Again, in vivo, the beneficial of in vivo is you can test all the secondary risk factors, challenging the mice to diabetic, challenging the mice to hypertension, so you can study all those secondary risk factors using the mouse models. The experimental technique, so all you have to have at least echocardiography so that you can measure the wall thickness, cardiac function, and if you have MRI, some institutions in the States they have MRI, they can also look at the mouse heart to see the fibrosis and contractile function as well. Then we have the pressure volume loop that also can measure the contractile, all the fine parameters of contractility. We can do histology, fibrosis, myocardial disarray, and then we can isolate RNA, protein, look single cell RNA you can do, single cell proteomics you can do. So a lot of things you can do from these mouse hearts. There is a very nice article this year, and I will send you this paper. This is in 2025 April. They covered experimental models of hypertrophic cardiomyopathy, a systematic review, a very nice review. You can see it's almost 30 pages. So again, it's a very tiny figure, but maybe here you can see. So the studies using mouse models, they compare from 1995 to 1999 to all the way. I think 2010 to 2014 has more number of papers using mouse models. Gene of interest, myosin heavy chain, already 70 articles been published with the mouse model, and then second one 60 with myosin binding protein C, titin 2, troponin C, troponin I, and then troponin alpha tropomyosin, then other sarcomeric proteins. Here countries of publications, you can see most of them from the States, and then also from China, some from Australia, and then the European countries. I don't see anything from India here. There are some studies.
For example.
Okay. So I'm sure, but maybe very small, couple of papers probably. The mouse model sarcomere gene is the predominant 80%, and the rest of them are non-sarcomere or compound heterozygous etc. combination. Again, the mouse models being used, hypertrophy, for example, if there is a model, what is the percentage in hypertrophic or the phenotype? Yes, here for example myosin heavy chain, a large number, myosin binding protein C large number, but there is a percentage have no phenotype. So again, when you generate a mouse model, it doesn't mean you will see a phenotype. It depends on the mutation, depends on where the mutation located in the gene, whether it's impact contractile function or not. Sometimes you don't see any differences between the wild type. So what you have to do is you have to challenge the mice to some cardiovascular stress, and then you can see the differences. So again, you have to play around if you don't see a phenotype at baseline. Maybe you can age them, you can see up to 1 year during the aging process, maybe you will see a phenotype. Sometimes some mouse models, they can go up to 1.5 years, 2 years, meaning that you have to spend a lot of money to pay for the colony, the cage. So in our university, per cage is per day is 75 cents. We can have up to 4 mice in one cage. So I have almost 350 cages. So per day you can imagine how much money we pay. So sometimes you see the mice, they fight, so you have to separate them, you have to hold them one single cage, you have to pay for the same cost. Whether you have 1 mouse or 3 mice, 4 mice, the cost is the same. They calculate based on number of cages, not number of mice. So again, these are the very nice articles I can send you. You can read more about the importance of the in vivo models and what are the models are in the literature and etc. So I will give you some examples from our lab. So this particular promoter, alpha myosin heavy chain promoter, we used to overexpress gene of interest in mice. So the question is, why do we use alpha myosin heavy chain promoter for a transgenic model instead of beta myosin heavy chain gene? So we have 2 myosin heavy chain, right? Alpha and beta. We use alpha in mice to overexpress the mutant protein or gene of interest. The question is why we don't use beta myosin heavy chain.
Strong promoter.
Okay.
Developmental stage. Developmental stage.
Developmental stage. Okay. So again we have 2 different paralogs, alpha and beta. In human, beta myosin heavy chain is the adult form. Alpha is an embryonic form. After birth we have beta myosin heavy chain. Before birth we have the alpha myosin heavy chain. In mouse it's just opposite. We have beta myosin heavy chain before birth in mice. After birth we have the alpha myosin heavy chain. So the switch occurs. Alpha myosin heavy chain is called faster myosin heavy chain because it hydrolyzes ATP very fast. Beta myosin heavy chain is a slow myosin heavy chain. It hydrolyzes ATP slowly compared to the alpha. Why this difference? So in mouse, as you know, in a minute it beats 500 to 600 times, so fast. So you have to have alpha to support that number of beats. In human it's 72 to 75, so we are switching, we are slower and we have the beta myosin heavy chain, and so that's why. So if you have a mutation in beta myosin heavy chain, you develop hypertrophic cardiomyopathy, but if you have a alpha mutation in alpha myosin heavy chain, mostly congenital and then also early dilated cardiomyopathy or heart failure, because alpha don't express as after birth, right? So that's the reason we have to use alpha myosin heavy chain promoter, and then we have a MIG-TAG. This is a 10 amino acid human tag. We put it in front of the cDNA of our interest so that we can differentiate the endogenous protein versus exogenous protein. So when you collect total lysate, how do you differentiate what is your transgenic protein versus your endogenous native protein, right? So this tag can be used. And then we have our gene of interest, and then we have the human growth hormone polyadenylation sequence. That sequence is critical to have a stable mRNA in the cells. Otherwise, the mRNA will get degraded very fast. So using this promoter, you inject into the embryo and then you generate the mouse. It's all the core facility. We do not do the entire process in the lab. We just outsource the core facility in the university. They generate the model and they give us the model, and then we start characterizing them. So again, this is an example. We published a paper in 2019. Expression of the mutant protein in the myocardium definitely cause hypertrophic cardiomyopathy phenotype, and so now we are using this model to test the gene correction or antisense oligos, whether we can reverse or improve the cardiac function. This is another example. People have now generated the pigs. These are the small, not the larger pig model. And so again, the myosin heavy chain gene was mutated. Here you have the beta myosin heavy chain, not in alpha myosin heavy chain. So again, mouse, rat, adult has the alpha, whereas rabbit and other upper animals, they also very close to human. They all have beta myosin heavy chain in the adult. So that's very good. But again, breeding rabbit or pigs or higher animals, more expensive, more time consuming process, and etc. And you don't have to have enough n number, you have to do a lot of breeding. But there are models. So this one, we published this paper with the Maine Coon cat. So it was very interesting story. So in the States, families, they own Maine Coon cat as a pet animal. You know, these Maine Coon cats are very large. They are really like a dog. You know, very big, and they have hypertrophic cardiomyopathy. Very natural selection. It's not artificially you introduce the mutation or not. And one of the families, they looked into the literature, who is here in this local studying hypertrophic cardiomyopathy. They found my name. And so they contacted me saying that their Maine Coon cat, through their veterinarian, saying it has hypertrophic cardiomyopathy and they want the cat to be treated. So they contacted me. I said, okay, I'm not a cardiologist, number one. Number two, I have to know the genetic cause for this disease. So I need to test whatever known mutations are. I can do it by simple PCR assay. I can decide, I can look, and then I can tell, okay, this is the mutation, this is a gene. That's all I can do, you know. They said, okay, we give you the blood sample, right? So we had to go through all the animal protocol, approval committee, and then material transfer agreement. You have to have everything. I mean, you know, these are very sensitive, issue. So if anything you do wrong, it goes to media and then you will be in trouble. So we got all the approval, and then we collected the blood sample, and then we tested for all the known gene mutations. There are not many for a Maine Coon cat. And then we found none. So we told them, this is probably the novel gene, novel mutation. All the known is not present in this Maine Coon cat. And they said, okay, how much would cost for you to do the whole genome sequence? Yeah, so I said, probably including bioinformatics, everything, 3,000, 2,000 to 3,000. Okay, we give you the money, please do it for us. Okay, then we performed the whole genome sequence of the Maine Coon cat, and then we identified the mutation in the troponin T. Then we said, okay, to conclude, we need the family. It's not, maybe it's sporadic, right? So then they went back to the breeder. The breeder is in a different city, and the breeder was unhappy because, you know, if they come to know they are using this model, then they cannot sell. it's bad news for them, but they were very coordinate, and they they identified all the siblings and parents, and you can see that how they are sister, and they come from here. The mother, the son is kind of the father of these two cats, so it's a very unique way they breed. Anyway, so this is a homozygous animal, so again it has a clear phenotype. So it was very nice story, but again this is the natural model. Now after seeing this publication, AstraZeneca contacted us saying, okay, we wanted to test our molecule in this model, so we are trying to collaborate now with AstraZeneca. So again, this is the way you kind of build your research. So we talked about hypertrophic cardiomyopathy versus heart failure. So in hypertrophic cardiomyopathy, you have increased force, increased contractile function, whereas in the dilated cardiomyopathy or the heart failure, you have decreased cardiac function or less force overall you generate. So here you have to use inhibition because you want to reduce the force generation, and here you have to use activator so that you can increase the force. So it's kind of two different way to treat hypertrophic cardiomyopathy. Again, we are going back to the sarcomere structure. We have titin, we have thick filament proteins, myosin heavy chain, myosin binding protein C, and then thin filaments, actin, troponin complex, and alpha trophomyosin. So myosin binding protein C we call as a brake, as you can see in this picture. It's tightly interact with the myosin heavy chain and control how much this interaction can be occurred so that the force can be generated. So this interaction is controlled by myosin binding protein C interactions. It can happen only when it's get phosphorylated. So myosin binding protein C gets phosphorylated only when you are hyperactive. Again, I told you, when you have increase in heart rate, this protein is highly phosphorylated, so that it goes away from myosin heavy chain. So the myosin heavy chain can interact with more actin and generate more force. So now so a lot of companies are now so much interested in studying these interactions and then modulate the interaction so that you can treat hypertrophic cardiomyopathy. Here again I want to show this is the myosin heavy chain protein, very large protein, 240 kDa. It has the essential light chain, regulatory light chain, different regions, and then the cardiac myosin binding protein C, all the interacting domains. So this is a study we performed in our lab. So we published a paper in JVC 2021 specifically in the S2 region. So most of the literature covered a lot of mutations in the head regions, but less interest was given to S2 region. This is the important region actually. When you enlarge that, you can see how many mutations being reported just from this region. These are the mutations are known causes hypertrophic cardiomyopathy. So we used, so again we have to use the animal model to study various secondary risk factors. So in this study what we try to come up with is identify the interacting peptide, the region, number one. Number two, how can we break or we introduce the break between the myosin S2 and then myosin binding protein C. One is phosphorylation, right? This is a dephosphorylation. When you phosphorylate C protein, that interaction is gone. So now we propose that can we introduce a peptide in between this interaction and can break the interaction. So we want to identify a small region of myosin S2, that exactly region that interact, and then use as a peptide therapy to break the interaction. So In heart failure, we have increased dephosphorylation. The more dephosphorylation you have, reduction in contractile force. So there are a lot of peptide therapies already in the market. So here I have some examples. Several diseases, they use proteins or peptides. So why not peptide therapies, right? So this is where Rohit, he tried to identify a small region of myosin S2, and that can be a potential peptide to treat either hypertrophic cardiomyopathy or the heart failure. So he used two different assays. One is a binding assay, another one is in vitro muscle assay. I will go through that one by one. So here when you express the recombinant protein and using HISTAC, you can purify the protein of interest in so clean. So again in bacteria you have so much protein, but you overexpress the mutant protein or protein of interest, and then using HISTAC column you can purify. You can see how enriched you can have. And then here you have the C0C2 recombinant protein. So this is the binding assay. You can coat the S2 and then you wash, and then you add the C0C2, then you wash, and then you put the antibody, and then you wash, and then you put the secondary antibody, and then you develop the color. So he developed a lot of peptides in overlapping peptides of myosin S2 region in collaboration with Amgen and the company. He found that these three peptides, 23005, 23006, and then 27119 show very nice concentration dependent decrease in the signal. So the more binding you have less signal, because the peptides compete with the C0C2, meaning that the C0C2 will be removed by washing. So and then you have less signal. So that's the way you calculate. Then we use, this is another major in vitro experiment we do, so called P-calcium, calcium force measurement, right? So we isolate papillary muscle from the heart, and then you can skin them. What does that mean is you can treat the papillary muscle with kind of detergents, so that you can remove the membrane, all the cellular components. You just have only the sarcomere proteins. So here you can see after the skinning, they are very transparent. You can see the striations actually very nicely. And then you can attach them into these clips, and then it goes into the motor, and then you can measure when these were in the small wells with a buffer. So you can increase the calcium concentration, and then you can see the force generation. So this is the sigmoidal curve. This is the maximum concentration calcium you need to generate this maximal force. So this is a very standard assay. Using that assay, you can look for how much force this can be generated. So here we have a PNAS paper where we showed that this is a wild type fiber, and then the wild type fibers were treated with entire S2 protein, and that can increase the force, maximal force. And then also we were able to determine how faster the rate of force redevelopment. When you stretch the muscle, how quickly it goes back and then come back again. You can see there is a significant increase. So it's kind of justification before we go for the peptide therapy. So Rohit used that approach, and he again selected these three peptides and measured the maximal force. Again he identified that the 23005 and 6 as well as 27119 shows very clear force increase. When we treat the fibers with these peptides, there is an increase in force. So how do we translate this peptide in vivo? So that's a question. So we have a model, mouse model, where we have mutated the phosphorylation sites to either through CRISPR-Cas9, either alanine mutation or aspartic acid mutation. So, this is the phosphoablation, this is the phosphomimetic. So, you can see here the phosphoablation show heart failure. They have reduction in cardiac function, enlarged heart chamber, etc., whereas the phosphomimetic is kind of normal in cardiac function. So, Rohit isolated papillary muscle from these animals. Here we have the wild type, then the heterozygous and homozygous of the phosphoablation, and then see that there is a reduction in maximal force of this fiber. Again, the phosphoablation means you have dephosphorylation of C protein. That means it has more break on the myosin heavy chain and less force being generated. So, using that model, we treat in vitro again and then challenge the fibers with S2 protein and then see whether that can rescue the phenotype. Again, we were able to see that compared to the control, treating the fiber with the S2 protein, there is a significant improvement in the force development. So, you can break the interaction of myosin S2 and myosin binding protein C using the recombinant proteins. Again, these are the beneficial of using mouse model or in vivo model. You can validate any drug development if you want to do. So, going back to the mutation story, so we have a paper in JBC how the S2 mutations that can impact the interaction with the cardiac myosin binding protein C and then cause hypertrophic cardiomyopathy. So, again I showed there are a lot of mutations in this region. We selected 2 mutations, one is a missense mutation, another one is a deletion mutation, and then see how that impact contractile function and developing hypertrophic cardiomyopathy. So, we have now a mouse model using CRISPR-Cas9 approach, a knock-in model, introduced the glutamic acid 903 deletion, which is in human beta myosin heavy chain, whereas in mouse we have it in alpha myosin heavy chain. The amino acid sequence is the glutamic acid 932, and we removed that as a deletion. So, that is exactly this region, and in families with this mutation is pathogenic, they show very severe disease, and so we selected this particular mutation. And here is our mouse model. These are within 6 months to 9 months of age. The deletion in myosin S2 region causes hypertrophic cardiomyopathy phenotype. They develop increased in heart weight, body weight ratio, and they also started developing heart failure after 6 months of age. Clearly, there is a reduction in cardiac function. We also have cardiac organoids, so for the same mutation, but in the beta myosin heavy chain, because this is from the human iPSC line, but isogenic line. Again, it shows there is an increase in contractile function, kind of replicating the phenotype of hypertrophic cardiomyopathy. From the mouse heart, we can do RNA-seq. This is Kalyani, my postdoc. She performed the RNA-seq. Again, you can see a lot of hypertrophic genes are upregulated in the heterozygous mouse heart compared to the wild type or non-transgenic mouse heart. This is a graduate student. She started this project. She validated this RNA-seq by doing RT-PCR individually on these genes, confirming that they are true. The RNA-seq is correct, validated, and there is an increase in hypertrophic markers. We also isolated the myocytes from this mouse heart. The myocytes can be skinned, removed the membrane, everything, and again you can challenge them to various calcium concentration. You can measure the force, how much the myocytes can generate. Again, you can see compared to the wild type, the mutant myofibrils show more force compared to wild type. So, here is a summary of this maximal force, and that is very significantly different. We also can isolate the fresh cardiomyocytes. These are not skinned cardiomyocytes. You can, once you isolate these cardiomyocytes are live within a day, you can measure contractile force and calcium transients. Again, comparing to the wild type, there is an increase in cell shortening, and then also there is a relaxation velocity also decreased here. So overall, these mouse models are being used to verify and validate the development of hypertrophic cardiomyopathy, and also use the iPSC's in vitro models to crosstalk. Again, I indicated that if you want to test the presence of secondary risk factors, there are a lot of ways to do that. So for example, if you want to introduce pressure overload, we can perform surgical model, aortic constriction, that can generate partial occlusion of the artery, and then the ascending aorta. So and then you can develop more pressure in the heart myocardium, and that can cause hypertrophy. We can induce a myocardial infarction, ischemia, reperfusion injury. This is the model which is very close to human, the reperfusion model. And then toxic, we can inject doxorubicin or isoprotrinol, angiotensin, etc., and then see how that toxic condition cause the heart failure. Or crossing the mouse model with another mouse model carrying a mutation, so you have a double mutation, how that can impact. Here is an example, like this was my PhD work. I was doing surgery on rat inducing pressure overload induced heart failure. It's a very standard technique. You partially occlude the aorta, resulting that you put more pressure into the myocardium, and that could develop hypertrophy within 8 to 12 weeks of time. So what are the challenges in the mouse model or animal model? Again, I told you, every model there are challenges. There is no perfect model, but you have to take the best suitable model for you to test the hypothesis, to develop therapies, etc. So again, even though we use a lot of mouse model, it's not the perfect 100% mimicking human model. There are a lot of differences and variabilities in penetrance. Again, mouse cardiac function, mouse, the contractility is entirely different from human. And then now there are ethical concerns in larger animals, so we have to really work on mouse or rats, etc. The primate model would be very good, but a lot of concerns on ethnics, etc. So in conclusion, in vivo models are critical, important for hypertrophic cardiomyopathy research, specifically studying the genetic mutations. And again, we should consider various types of models, and comparing to in vitro, we need to integrate all the technique so that we can compare and validate all our findings are accurate. So the future ongoings are like how can we introduce gene editing or mutation within embryo, so you can correct the mutation before the baby is born, or so we can correct the mutation very early on. So final summary is animal models are key for various purposes. But again, it's not the perfect model, 100% not perfect model, but it's important to choose the right animal model based on the question you are asking. So your next mouse model is only a mouse click away, so you don't need to do it, you just pay for it. So a lot of companies, a lot of core facilities, they just do it for you. It's a lot of time, I mean, it's time consuming, and these days it's cheaper actually compared to 10-15 years ago. With that, I want to acknowledge some of my lab members, specifically James, Rohit Singh, and Brendan from Amgen. He was one of our collaborators. They provided also funding support to characterize the myopeptides. So with that, I want to conclude and this session in vivo, and happy to take any question you have, and then we will have a break for a short break for tea. Thank you. So animal model, in vivo model, why, how, what is your thought, what is your question? Yes.
Question with regard to the peptide therapy.
Yeah.
Yeah. I mean, just technical question. What or how did you decide the amino acid residue sequences in the peptide therapy to use those peptides to apply it as a break on myosin?
Yeah, so you take a region of the binding, right? You take protein A, protein B, and these two protein binds here. So you take this region, right? And then you generate overlapping peptides, 10, 15 amino acid, 10, 15 amino acid. So you are making overlapping 50 peptides. Okay, and then you narrow down which are the peptides are really binding.
Okay, so initially a combination of the peptides were tried,
yes.
and then based on the data.
Yes.
Okay.
So once you narrow down, then for example for the in vivo purpose, you have to attach homing peptide, another peptide that is very specific to cardiomyocyte. So once you put the peptide into the bloodstream, this goes into circulation. How do you deliver this very specifically to the cells, to the heart cells? Otherwise you will be cleared in your liver and it's gone, you know, or it will go to skeletal muscle randomly somewhere, right? So you add homing peptide so that you can deliver these peptides to specific cell type.
Okay, so
But we are not at that stage yet.
okay, yeah, got it.
Have you taken any help from in silico simulation for this selection of the peptides?
Yes, we have done it. I don't have, I will show you the data maybe tomorrow. I have slides.
Okay.
We narrowed down to 3 amino acid through that model.
Okay.
And then we use that model to identify small molecules. That molecules also can bind similar to the peptide. So instead of the peptide now you can put the molecule and that can do the same job as the peptide.
Okay, and that is how you ask Amgen to provide those peptides, correct?
Amgen helped in the peptide project, but again it's you have a 1 year contract, 2 year contract, and now it's over. So now we have they opt out from the intellectual property, but we have the person from the Amgen, Aaron, he is part of our project. So we have the provisional patent. Now we have identified 12 small molecules that can mimic this peptide. So now we have to test these small molecules and then we narrow down and then we can go to the next patent.
And these peptides were designed to reduce the kinases activity, correct? Phosphorylation.
Yeah, so the peptide is to break the interaction between myosin heavy chain and myosin binding protein C, particularly when you have a decrease in force, because the C protein puts more brake on myosin heavy chain. So you want to put the myosin S2 peptide that competes with the endogenous myosin heavy chain and then release the endogenous interaction. Then you have the endogenous myosin have more actomyosin interaction, generate more force.
Thank you, sir.
Questions from students? Any questions from the students, master students? Yes.
Toxic cardiomyopathy, and in front you had written dox.
Doxorubicin.
Doxorubicin. So doxorubicin
it's a cancer drug.
Yeah, it's a cancer drug. So,
Yeah.
so administration of this drug is, I mean, can exert this kind of
Correct. Doxorubicin can cause dilated cardiomyopathy. Most of the cancer people, they die not because of cancer, it's because of the heart failure, because of the drug.
So that is what is the side effect of the
Yeah,
drug.
side effect, yeah. So it's a lot of work is going on having cardioprotective cancer drug to overcome doxorubicin, but it's still the best drug in the market. I think Princess has a question. For students, if you ask one question, I give you 100 rupees per question.
I think maybe I can wait for
No,
the
you can
students
ask,
to ask.
I just give some time.
Okay, so about this,
100 rupees per question from students, okay?
about this alpha to beta switch, like beta to alpha switch or whatever in both the organism. So, like I have two questions. So one is the based on the rate, like you said in rat we have a reverse thing, like beta first and then alpha in adults, and in the human we have vice versa. So what about the highest like the elephant and other things? Do they have any other form of some like gamma,
I
or
don't
it is
know.
not discovered yet?
I don't know, yeah.
Okay.
Probably, I don't know.
Okay.
I don't know what elephants, adult elephant.
Like that is also 5 times lesser the heartbeat.
Yeah,
17 something.
yeah,
So
so we will see.
and other one.
We have here, because the
Yeah,
Department of Zoology,
genetics, yes.
I'm asking the professors, is there a possibility to have elephant heart samples? So you can isolate RNA,
I think there's a tissue sample.
I mean, elephant
Small tissues.
tissue. When they naturally die, you can work with the forest officers, I mean, naturally dying elephant, you can collect the heart, right?
Yeah.
I mean, you preserve the samples, everything.
Yes,
You can take some elephant, naturally die. You are not asking any elephants or you are killing it, When they naturally die.
But the collection of samples may take years.
Yeah, you have to set everything and wait for one day, you get one sample, then you get plenty of data. Yeah.
just one more small question. So about this alpha to beta in human, the switch occurs in the very beginning during the developmental process. So by chance, can we control the switch? So like suppose we know that okay,
You can control the switch at the transcription level.
Yes. Like suppose C protein has a lot of mutation and that is causing a lot of problem. So can we switch the expression of alpha to beta when it is in adult form by some transcription factor, and that can accommodate those faulty
Yes.
C protein?
Yeah.
I mean, is it
Yes,
can it be
so,
done?
so for example, the technology called CRISPR-A, the new technique.
Okay.
This CRISPR-A can go and bind to the promoter of interest, activate the transcription.
Okay.
So
Like-
it is possible.
Like suppose like there is a competition between the peptide you inserted, and then like suppose alpha is also equally expressing. Even the rate will be higher, but at
Yeah.
least there will be something to
Yeah,
compete with or
100%.
complement with.
Yeah, there is a possibility.
Okay.
Yeah, good question. There is one, the last. He's not a student, but
Sir, after mutation, how you overcome the natural repair mechanism? After inducing the mutation in mice, in particular peptide, how you overcome the natural repair mechanism?
how do you overcome the So again, treatments is mostly for the improvement or try to rescue as much as can. If you can improve a quality of life of a patient, 1 year, 5 year, 10 year, anything today is better, right? So you are working towards that kind of goal, but not completely reverse, because you still have the mutation. You born with the mutation, you have the mutation, and so either you have to correct it completely at one early stage, or before birth or after birth in a quick time. Otherwise, you have the mutation forever, so you have to have a permanent treatment option. once you have the phenotype. Yeah. Questions from master students? Not from you. You can ask. Yes, anybody in the back seat? Otherwise, I'm going to pick up your name. I have all the names here.
So you said in humans, before birth we have alpha myosin, after birth we have beta myosin.
Correct.
So the genes that express alpha myosin must be switched off after the birth
Yeah.
through some kind of mechanism like
Yeah, transcription
decapping
factors,
or.
yeah.
Is there any mutation in which that mechanism gets turned off and both alpha and beta myosin express simultaneously?
I don't know, it is possible. If the transcription factor failed to switch, right, then there is a possibility, but I don't know any literature top of my head.
Sir, and one more question. You showed the pictures of animal models that are used for
Yeah,
study of the disease. You showed the picture of a chimpanzee, which is a very higher order primate.
yeah.
Is that
So-
actually used in the study?
Yeah, they use for hypertension, some natural, cardiovascular diseases. So in Germany, in some places they have the primate facility. They study some cardiovascular diseases.
Because they are endangered animals and
Yeah.
also quite big, and
Yeah,
they can pose threat to the humans. So
I don't know how they maintain that, but possible. Okay, let's have a tea break and then we will meet in 10 minutes.
Thank you so much, sir. This was really an interesting talk, the way you explained with all the pictures and videos. And the most interesting part which I and audiences is the organoid structure part. That is something new.
Thank you.
So yeah, definitely looking forward to learn from you. And yes, go for a coffee and we will meet in 15 minutes. Thank you.